to show you how to collect water and zooplankton samples in deeper water bodies. This in particular applies to the freshwater tidal and lake ecosystems. And so we don't use any special water collection tubes or devices. We simply use a sump pump, which is very similar to one that you would put under your house, and vacuum tubes, which these are sort of old-style vacuum cleaner tubes, and a mark roped with deck. So we essentially bring a 12-volt battery from a car. We connect that up to the sump pump to turn it on, and then we start lowering the tube. Sorry. And the sump pump, and we collect water at the surface and multiple depths in the lake. In the river, we usually collect water in the top, middle, and bottom thirds. In most habitats, we have some flow even in the lake, and so in order to keep the device up and down vertical, we attach a mushroom anchor to try and keep it in one place. And so we basically turn the sump pump on and clear the tube, the vacuum tube out, and water comes out of the end of this vacuum tube, and we put that into a collection bottle, a 1 liter collection bottle, and then we prep it for filtering. For zooplankton, we continue at each depth, so we successively collect water and then zooplankton. For the zooplankton, we use just a regular old 5 liter or multi-liter, 5-gallon, 16 liters bucket that you can get from any hardware store, and we use some large PVC pipe, and on the bottom, you can see that better, is a 44 micrometer filter. And so we use a filter so that we can capture zooplankton, which are microscopic critters, and we want to filter quite a bit of water, usually 32 to almost 100 liters of water, depending on the density that we're seeing. And so we record the volume of water that we filter. We essentially take the water, so imagine that we're at a location. We take the water that's coming out of my tube, coming from my sump pump, we put that into the filter, and we filter probably 2 to 4 buckets of water, and in between we have to dump buckets. If the filter gets a little clogged, we have to find a mechanism to unclog it. So then we've got no water in here, we've just got a bunch of organisms on top of the filter. So we've captured our zooplankton on our 4 micron filter, and we have to get them off, and we have to preserve them. And so the way we do that, and this is takes usually a couple students, we use the 1 liter collection bottle, and then since the zooplankton are on that surface, we use a funnel. We invert the collection tube, and we squirt the zooplankton off, and they go down into the bottle. And then we preserve with a solution of ethanol and usually a little dye to help color the zooplankton so that we can see them better under the microscope. Okay, we also collect water in 1 liter bottles, and then we filter it in the field. And we filter it for both chemistry or chemical tests that we'll conduct in the lab, and then we filter just for chlorophyll A, and we only need the filter for that. So what we're doing when we filter is we're trying to separate the organic matter from the inorganic matter that is dissolved in the water. And so we don't want any more electricity out on the boat than we have to have, and so we use just a regular manual hand pump. So this is the filter that I prepped earlier. Grab it. Microfiber filter that will have, once I've filtered water through it from the field, it will have sediment and other solid elements on it. And so that goes on the top of a filter pump. I pour my sample, which I have a bottle that I have my sample in. I pour that into the graduated cylinder, and I filter a known volume of water, usually 250 milliliters for total suspended solids. Pour the water into this, and then I pump under pressure, and then that water that goes into the bottom of the speaker will be filtered water and will go into another smaller sampling container, 250 milliliters. And then I also save off another 250 milliliters of unfiltered water. Rinse the entire apparatus. Remove the filter after filtering and put it back into the foil. And you have to be very methodical, no half steps in this. If I accidentally didn't put it back into the filter, or I didn't close the filter, I might destroy the sample. So we want to make sure to put it back. We use another type of filter, a Metrocell membrane filter, for filtering water for chlorophyll A. See those filters, they have little dividers in them. Okay, and same with this filter. It goes on the edge of my finger for this. Love showing this. Okay. And same setup with this filter. You're just going to filter 25 milliliters and save the filter. When we get back to the lab, we put these in the freezer so that there isn't any decomposition of the chlorophyll A.